Hello, my name is Landy, and I'm a fifth-year Ph.D. candidate at Northwestern University. I work in the Department of Chemistry under the guidance of Professor Chad Morgan. Before coming to Northwestern, I got my bachelor's degree in chemistry from the University of Minnesota, with minors in sustainability studies and biochemistry. Research thrusts in the Merkin lab focus on preparing new materials with nanoparticles as building blocks. By definition, nanoparticles have at least one dimension between 1 and 100 nanometers in size. Because of their small size, nanoparticles have unusual properties. For example, a solution of gold nanoparticles can look red in color, very different from the familiar shiny yellow color of gold bars, rings, and metals. You may also know that diamond and graphite, or pencil lead, are both made of carbon atoms in different arrangements, and that, as a result, these materials have vastly different properties. Just like atoms are the building blocks of conventional matter, nanoparticles can also be used as building blocks to build larger and more complex materials. These materials also have different properties based on how their fundamental building blocks, the nanoparticles, are arranged. But how do you hold nanoparticles, something so tiny, together? What we need is a special type of glue. DNA is often called the blueprint for life because it contains the instructions in the form of a chemical code needed to make proteins vital for many cellular processes and functions. In our lab, DNA is used as a chemical blueprint and bonding agent to glue together nanoparticles and program the formation of larger crystalline assemblies. We use an instrument called a DNA synthesizer to make custom DNA strands with different lengths and sequences, where the sequence can be used to deliberately program how strands match up and link together through A and T and C and G base pairs. One can covalently attach many DNA strands to the surface of nanoparticles of a variety of sizes or compositions, resulting in a dense shell of DNA oriented outward from the particle core or atom. When nanoparticles bearing complementary DNA shells are mixed, the DNA strands between neighboring nanoparticles bond with one another, carrying the particles along with them to produce extended arrays of nanoparticles that we call colloidal crystals. Traditional atoms interact with one another to form molecules and materials through electronic bonding involving their valence electrons. And the DNA-modified nanoparticles that I just mentioned interact with one another to form materials through molecular DNA bonds. In this way, they can be likened to atoms and are known as programmable atom equivalents, or PAEs. For more information about PAE nanostructures, please watch this accompanying video. And the similarities continue. Another important concept in atomic bonding that you may be familiar with is the octet rule. This rule states that atoms are stable when they have 8 valence electrons, so they prefer to form bonds until they reach this configuration. PAE systems also seek to reach the most stable configuration of bonds, but here the most stable configuration maximizes the number of DNA hybridization events between neighboring nanoparticles. The complementary contact model, or CCM, was developed based on this observation to help understand and predict PAE bonding. It is analogous to the octet rule, but ultimately more powerful for building materials by design. The CCM was originally developed in the context of spherical particles, but it can also be used to predict the PAE geometry when particles of different shapes are used. If we use a flat, disc-shaped nanoparticle core, for example, instead of a spherical one, the PAEs can maximize the number of DNA bonds between circular faces by aligning into 1D stacks. Cube-shaped nanoparticles also have predictable bonding arrangements based on the CCM, where 6 cubes can maximize their DNA bonding by aligning with the faces of a central cube. This arrangement is much like the familiar octahedral geometry of a sulfur hexafluoride molecule. When each nanoparticle cube is surrounded by other cubes in this geometry, the extended lattice is a simple cubic structure. Proteins are another exciting building block that can enable new directional bonding approaches. DNA can be attached in atomically precise locations on their surfaces, offering immense tunability in bond distributions. Another concept you may remember hearing about in general chemistry is valence shell electron pair repulsion, or VSEPR theory. In VSEPR theory, the three-dimensional coordination geometry of an atom is defined by the arrangement of electron pairs in a way that minimizes repulsion between them. A similar concept arises when we consider very small, sparsely functionalized particles that can act as electron equivalents. Now, unlike PAEs, which adopt fixed positions within lattices, electron equivalents can roam through the lattices of larger PAEs, similar to the sea of electrons that describes metallic bonding. The strength of interaction between larger PAEs and electron equivalents dictates their arrangement relative to one another. With spherical PAE systems, the DNA bonding is symmetrical in all directions. These electron equivalents can break this symmetry to enable directional bonding with otherwise isotropic structures. Despite these similarities, there are also several key differences between atoms and PAEs. Unlike traditional atoms, where the bonding characteristics are fixed by nature and atom identity, the bonding characteristics of PAEs are independent of particle composition and depend on DNA position, sequence, and length, as well as nanoparticle size and shape, as determined by the researcher. Also, unlike with atoms, where an atom's identity and how it bonds are linked, researchers can design and synthesize PAEs with DNA bonds and nanoparticle pores independently from one another. Colloidal crystals can be prepared that have the same structure, but different nanoparticle pores, and we can separately tune bond length and strength. These differences offer exciting opportunities to design and prepare novel multi-component materials using PAEs. We can even make structures so unusual that they have no known mineral equivalent. Because of their nanoscale size, these crystals interact with light in interesting ways, making them useful in the development of optical devices. For example, colloidal crystals can act as nanoscale versions of lenses and mirrors that can focus or reflect certain wavelengths of light, and different regions of the spectrum can be accessed by tuning the properties of the building blocks. The chemical responsivity of the DNA shell can also be exploited to dynamically tune the optical properties of these assemblies. Ultimately, colloidal crystals could be used to permit the development of nanoscale devices integral to photonic integrated circuits, or computers that use light instead of electricity. Now, in my own research, I am building materials that are similar to substitutional alloys of metals using PAEs. Using the DNA shell design, I can program how two PAEs with different nanoparticle pores can substitute into an ordered or a random alloy structure. My goal is to continue to enrich the table of nanoscale PAEs and draw analogies between PAEs and DNA bonds and atoms and conventional bonds. I love doing research because it challenges me to ask important questions, to solve problems creatively, and contribute to the broader scientific community. I started doing research for course credits as an undergraduate, and my grad student and faculty mentors became an invaluable resource for me to help apply for scholarships, summer research funding, and eventually for graduate school here. Undergraduate research helped me develop skills that I use in graduate school and life every day. So I encourage anyone considering it to take advantage of the research opportunities at Northwestern and get involved in research right away. Thank you.